Hi guys, it's Dr. Orlando. Going to do a video on biomolecules. Now this is just going to be an overview. You know that you will find a lot more information in your lecture notes, in your textbook, and your online resources. This is meant to give you the big picture, to make sure that you understand the big picture, and then it's your job to fill in all the little extra details that we talked about in class. And it should be easier to fill in those details if you know where they belong. When you have the big picture in your mind, that's a little easier to do. Okay, so if you have any questions about the details, you can go ahead and email me or call me about them. All right, so we're going to talk about the four groups of biomolecules we talked about in class: carbohydrates, lipids, proteins, and nucleic acids. And we're going to talk about their function, and then we're going to talk about what their monomer looks like and what their polymer looks like. Okay, so first, function. Function of carbohydrates. Lots of functions, but we listed just a few of them here. So energy. We need, for example, we need glucose to burn to make energy. We also need the complex carbohydrates as storage of energy, like glycogen. Okay. We use carbohydrates for structurally in the body, for building things. And then we also use them as cell-to-cell signals, little messengers that go between cells, and we also use them as tags on cells so that they can identify the different types of cells and whether they belong to us or not. Okay, lipids. Lipids are for energy storage. So fat stores. Can use it for waterproofing, like our Labrador Retriever dogs and our ducks. Big time important function with cell membranes. And then hormones as well, some hormones. Okay. Alright, move this over. Protein function. I'm just going to put it out there. Everything. Everything is protein. And that is the job of a protein. Even if you want to make lipids, or you want to make carbohydrates, there are protein enzymes that do that. Okay, so enzymes is a huge job for proteins. Okay, enzymes, give that a star. Okay, proteins are structural. They are important for movement. Remember, I talked about the proteins that slide past each other to make our muscles shorten, muscle cells shorten. And then proteins can also be hormones. Our smaller little hormones are proteins, or peptides even, the short proteins. Antibodies that help us fight off infection. Antibodies, okay, help us fight off infections, our immune system. So they are proteins. Okay, function of nucleic acids. Big time job, of course, is to be our genetic material. Okay, and so this is our DNA, RNA. And then another job, and this one we drew a picture of ATP. You may recall nucleic acids can be energy carriers. Well, not nucleic acids. This is a job for nucleotides. Remember, nucleotides are the monomer. We're going to talk about this in just a second. And the bigger structures, when you hook them together, is the nucleic acid, and that's DNA or RNA. Okay, so let's go back over here to carbohydrates now that we have everybody's jobs done. And we're going to draw the line across there. Sorry, I hope this doesn't make you motion sick. Not sure if there's another way to do this. And we're going to move you up. Give me some drawing space, gonna need it. Okay. So now we're going to talk about the monomer of this. Okay, monomers. Okay, so monomer refers to the single subunit of the larger biomolecule. And I'm going to give you some examples of the monomers. I'm not going to give you all of them that I talked about in class. Don't have time for that. Again, it's your job to fill in the blanks, but I'll give you the most common example and the big one that I talked about in class. Okay, so monomers, this column, this first column here that we're working on is carbohydrates. I'm just going to write at the top here since we can't see our labels. The carbs. The monomer is the monosaccharide. A-R-I-D-E. Okay, monosaccharide. And that means one sugar. So it's a simple sugar. And the examples we saw in class were, some of them were, I gave you quite a list there, glucose, fructose, galactose. Okay. Notice the oses at the end of their name. That should immediately give you the idea that you're talking about simple sugars, monosaccharides. We will see other oses for the disaccharides, but at least we'll tell you you're talking about sugars and carbs. Okay. All right. So what do glucose, fructose, galactose? What do they look like? And let me add ribose in there so we can have one that looks like that too. Most of the time they're kind of a hexagon shape. They've got carbons sticking off of them. Right? Remember the bend, wherever there's a bend there's a carbon. And many times they have, and they will have this oxygen here in their bond. Making an ether bond there, or maybe they are a pentagon shape, oxygen at the top there. Okay. And that they always tend to make this ring structure when you put them in an aqueous solution, watery solution, and since we are a watery solution, this is really the form that you're going to see drawn in most biology textbooks. Okay, so that's the monomer for carbohydrates. All right. The monomer for lipids. Now this gets a little bit more complicated because these are more variable looking. Okay, so for lipids you're talking about a couple different groups. Remember there was fats, waxes, and oils, and their key was that they were all carbon, hydrogen, oxygen. Okay. So the example for fats, waxes, and oils that I gave you was a fatty acid. Okay. And the fatty acids, remember, they always have a carboxyl group. Okay. And then they are a chain of carbons surrounded by hydrogens. Remember, that's the zigzag line. So at every bend there's a carbon, and that carbon is surrounded by hydrogens, the way that I've drawn this. And at the end, there's a methyl group. Okay, so that's our fatty acid. If it's floating around by itself, you might call it a free fatty acid. Okay, then we had phospholipids. And phospholipids were always drawn like a circle with two tails. You can draw them like that, or maybe better that I do this way. Hold on. Let me do a redraw here. Let's draw our zigzag lines in so that we remember this is actually a fatty acid tail. So you've got a fatty acid there and a fatty acid there. So you might not consider this truly a monomer since it has more than one fatty acid, but it kind of has a job by itself like a monomer, so we're going to draw them under monomers. So the phospholipid has a head that is hydrophilic, and tails that are hydrophobic. Okay. And so the phospholipids, as you may recall, they have a job of creating our cell membranes. Okay, remember the phospholipid bilayer. I'm going to write that down because I want you to make sure you remember that. And you should definitely, if that doesn't immediately bring a picture to your mind, you need to go straight to your lecture notes or textbook and read about phospholipids and how they create the phospholipid bilayer, because that is critical that you understand that. Okay, I'm going to move up a little bit because lipids need a little bit more space. The last lipid that I'm going to talk about is cholesterol. Cholesterol. Steroids / cholesterol. Okay, remember, cholesterol, I don't expect you to be able to draw cholesterol, but it looks like this. It's got 4 rings. Like that. And there are side groups off of these rings. So it looks different than the other lipids. There's no fatty acid tails here. Okay, but cholesterol, the sterol will tell you that you're looking at the steroids. So ones like estrogen, testosterone, cortisol, aldosterone. Okay, so those are examples of your monomers of lipids. Alright, this way and this way. And now we're going to talk about proteins. Oops. Okay, so the monomer proteins are pretty simple. You have the amino acids. And amino acids, there are 20 different amino acids, but they have a basic structure that you have to know and be able to draw and describe. So it looks like this. There's a central carbon. It's got a carboxyl group. Carbon double bonded to oxygen. An OH, hydroxyl, and then it has an amino group, NH2. Okay, and then they all have a hydrogen side group. Oops, talking and writing. All have a hydrogen side group, and then they all have, and I'm going to give this guy a different color. They all have an R group and give them a star. So the R group, you hopefully remember that the R group is the variable group. Anytime we write a letter R attached to one of these basic chemical structures, we know that that means that spot varies in what is attached there. So depending on which amino acid you're looking at, that R group is different. So some of the R groups are hydrophilic, some are hydrophobic, okay, have charges, acidic, basic. So they've got characteristics that give those amino acids unique properties. Okay, so those are the monomers of proteins. All right, last up is the monomers of nucleic acids. Okay, and for nucleic acids, again, you have to be able to draw the basic structure of a nucleotide. is the monomer, and remember it has a sugar, which is going to be like that, and that sugar will always be deoxyribose or ribose. Os tells you sugar. And this is just one of those monomers that has another biomolecule as part of its monomer. Okay, so a nucleotide has a sugar. It also has a phosphate group, at least one phosphate group, and it has a base. I'll just put the letter B there. Remember, this base, there are only 5 possibilities for the base, what they can be, and I'm going to just put their abbreviations here. I'll say what they are. So adenine, guanine, cytosine, thymine, and uracil. Okay. So that base could be any one of those bases, depending on whether you're looking at DNA or RNA, or what kind of nucleotide you're trying to make. Okay, the other thing that I want you to know about nucleotides, and I drew this picture as well in class. So this would be the first picture would be a good drawing for just your basic nucleotide that would go into DNA. Now, some nucleotides have jobs, as you know, have jobs just by themselves. They're not attached to a big strand of DNA. They have a job. And that one that is super important is ATP. And that is adenosine triphosphate. And I put that dash in there because I needed the room. It doesn't exist in a normal, if you're just writing it straight across. Okay, so let me draw ATP real quick. Basically, you're drawing what you just drew above. So you've got your sugar. The base is adenine. Okay. And the phosphate varies just a little bit. There is a phosphate, but guess what? This is a triphosphate, so he gets 3. And why is he so important? This bond right here, super important. Remember, when we break that bond, we get energy. Okay. So ATP carries energy in the form of that covalent bond right there. And we will do a whole chapter on making ATP for energy. Okay, so that is our monomers. Okay, I'm going to go to the next page for polymer. Okay, guys, I'm going to talk about the polymers now. So we'll start with carbohydrates. And under carbohydrates, we talked about disaccharides. So this is where we joined 2 monos. An example was glucose plus galactose yield lactose, which is milk sugar. Okay. Then we talked about polysaccharides. Okay. And under polysaccharides, we talked about glycogen. Remember, glycogen is the storage form of glucose in animals, stored in the liver and muscles. Starch, storage form of glucose in plants. Cellulose, the most abundant of the polysaccharides, and it makes the hard outer protective covering of cell walls. It's what makes trees strong. Okay, and then last, chitin. Chitin is a unique polysaccharide in that it contains nitrogen, And we find that in the exoskeletons of insects, in the shells of crabs and lobsters and other crustaceans. We also find it in the cell walls of our fungus, mushrooms. Okay. All right, on to lipid polymers. Okay, so for lipids, the example I used in class is the one I'll use now, and that is a triglyceride. So for triglycerides, remember that triglycerides, the glycer part of it, is referring to this 3-carbon backbone known as glycerol. Okay. And then attached to them is 3 fatty acid tails. And we don't need to add in the methyl at the end when you're drawing these. It's known. This would be the methyl end. This is where the carboxyl used to be. Excuse me. Now it has been dehydrated. It's gone through a dehydration process. Okay, so we can put where carboxyl used to be to give you orientation. All right, and triglycerides are an important energy storage. They're important for energy storage because they have so many covalently bonded carbons. We can make a whole lot of sugar out of those, and if we can make sugar out of them, then we can make ATP out of them. Okay, we can also burn fatty acids and glycerols. So just think of all those covalent bonds and all the energy that must be stored in them. So you want to think this is where they are. They're found in fat stores, and they are for energy. All right, proteins. For proteins, the polymer, a small polymer in proteins would be called a peptide. And we would say this is less than 50 amino acids. That's pretty short. So remember, amino acids are the building blocks. So we connect them together. There's no reason that one is any longer just because of my drawing isn't great. Okay. But it works out well because then I can point to the bonds between the amino acids and ask you, what do you call that bond between amino acids? We call that the peptide bond. Okay? All right. And if we have greater than 50 amino acids, we call that a protein. And with respect to proteins, there was quite a bit of information on proteins if you go back through your notes, but remember, I want you to be sure to understand protein structure. Because with proteins, their three-dimensional shape is everything. Okay, what gives them their shape gives them their function. So anything that might happen to change that shape, like a change in the environment, for example, can be a problem, can be good or bad for that protein. So make sure that you go through that. Remember, that's that primary, secondary, tertiary, and quaternary structures, okay, of protein. So make sure you know that. All right. Lastly, polymers, nucleic acids. You're looking at DNA and RNA. Okay, and for those, I'm going to draw DNA. Okay, just a generic DNA here. Remember that the phosphate of one nucleotide connects to the sugar of the next nucleotide. So remember, I said DNA was like a ladder. Okay. Of course, we twist that ladder to give it the exact right shape. But in our ladder, the rails, or the sides of the ladder, are that phosphate sugar. Okay, we could go on. And we can put circles around these too. I think I'm going to zoom in a little bit here. Easier. If you're drawing this and you use different colors, sometimes that looks really nice too. I haven't found a fill tool yet on this. I bet there is. There's probably on here a fill tool to where I can just fill in these circles. Alright, so those are the rails of the ladder. Okay, I'm going to draw one half at a time. And then what makes a nucleotide, just draw one nucleotide over here. The subunit, remember, in my little key so you understand what it is. We'd have a sugar, a phosphate, and a base. Okay, so that's one nucleotide. So we've got the sugar, got the phosphate, let's add a base. We're just going to put B. We're not going to worry about base pairing in this lecture, so we're just going to use the letter B. Sorry about that. Notice how I'm making sure that my bases are connected to my sugars, because that's how it really, really is. Okay. So then over on this side, this is DNA, deoxyribonucleic acid, which is a double-stranded form of genetic material. So it does look like a ladder, because it has two strands. This is half of a strand. So we would draw the same thing on this side. Phosphate. I want to make sure that my sugars really end up across from each other, so I'm going to put them in first. Otherwise, when you end up drawing, it gets all cattywampus. I like that word. Okay. Okay. So across from attached to the sugar, we're going to put a base. I know I made these a little bit smaller. Don't sweat that. They really should be, I mean, as far as size goes, this should be a symmetrical looking structure. These bases are not identical to each other. They are definitely not identical to each other. They're complementary. So in the end, let's say the width, if we were to measure, let me grab a little color here, if we were to measure side to side like that, the rails of our ladder, the rungs of our ladder would all be the same width. So those would be the rungs. Okay, so in my ladder analogy, the rails of our ladder are the phosphates and the sugars, and the rungs of the ladder are the bases. Okay, and we can add in some little dots. You guys remember what the little dots mean, right? Those little dots mean weak attraction, but it's pretty strong because it does cause this entire thing to spiral. It's one of the things here. Those hydrogen bonds. Okay, hydrogen bonds, hang on a minute. Hydrogen bonds, weak electrical attractions that actually will create the spiral. Okay, so you'll hear DNA referred to as a double helix. So in the end, you take your ladder and you give it a twist, just like that. That's DNA. If we were going to draw RNA, we would just draw half of that. Okay? We draw one rail and a half a rung. Half rungs. Okay, I think that's everything there. Boy, that's a half an hour. Okay, make some time to listen to this. And if you've got all of that down, go fill in the blanks. And make sure you get all the rest of the details that I talked about in class. And if you have any questions, please feel free to call me or email me. Hi guys, I just wanted to fix a mistake that I just saw in here when I'm talking and drawing at the same time. I'm going to fix my carboxyl group here Yeah, Dr. Orlando. C double bond oxygen, OH. That's a carboxyl group. Just like I drew over here on our amino acid. So, just want to make sure, keeping myself honest here. That's it. Thanks.